Hello everyone. In this video, we will see what is apoptosis, how and when it occurs, and what is its physiological significance. So first let's see when the cells can die. The cells can die either when they are infected or injured or poisoned. In these cases, there is release of toxic substances from the cells which actually acts on the surrounding cells and it can cause inflammation. This kind of cell death is known as cell murder or necrosis because it is happening due to an external insult to the cell. On the other hand, physiologically, there is a balance between the survival and death of the cell. So, there are certain factors which promote survival, which we call as growth promoting factors, and if there is absence of those factors, survival may be affected. Similarly, the cell may receive a death signal. So, if the cells do not receive a life maintaining signal, the balance will shift towards the death of the cell. On the other hand, if the cell receive a death signal also, then also the balance will shift towards the death of the cell. So, this is known as programmed cell death and is considered physiological death. So, this programmed cell death is named as apoptosis, literally meaning falling off. We will see why it is known as falling off. And this is a cell suicide. This is not happening due to an external insult to the cell, but the cell, the program within the cell is causing it to generate a death signal, or if there is some other injury to the cells which release growth promoting factors maintaining life for these cells, then also there will be apoptosis. So, this occurs when there is a need for removal of unnecessary or damaged cells, and it is a physiological method by which cells die, and this process doesn't affect the surrounding cells. So, it is affecting the only cell, right, that receives a death signal or do not receive a life maintaining signal without affecting the surrounding cells. So, it is kind of a cell suicide. So, what is the significance of apoptosis? First, let's see its physiological significance. Apoptosis is very important during embryogenesis or during development. So, during development, what happens that lot of cells start dividing and basically are forming a mass of cells. So, for proper shape of the various parts of the body or organs, what happens that we want extra cells to be removed, isn't it? So, here is an example of webbing of fingers where extra cells in between the fingers need to be removed, right? So, this is one example during embryogenesis. Apoptosis is important. Secondly, apoptosis of self-reactive lymphocytes occurs during when lymphocytes are developing. Lot of lymphocytes develop which are reactive against our own self antigen. So, we should identify these lymphocytes. I mean, the body should be able to identify these lymphocytes when they are developing, and then there should be apoptosis of these self-reactive lymphocytes. Otherwise, it will be too harmful, isn't it? Then, it also regulates number of cells. How? One example is say in ovulation. In ovulation, what happens that during the development of follicles, lot of follicles start developing, but ultimately there is selection of one dominant follicle only and rest of the follicles undergo apoptosis. They are not needed. So, it is kind of regulating the number of cells also. Then, it does away with cells whose job is over. So, there is a particular lifespan of cells. So, when their lifespan is over, they undergo apoptosis. One example, say suppose in GIT intestinal epithelial cells, they undergo a rapid turnover. So, they are undergoing apoptosis as soon as their lifespan is over. Secondly, in menstruation also, say suppose during the menstrual cycle, we have the development of the uterine epithelium by the hormones estrogen and progesterone, right? So, that is in preparation of a fertilized ovum. If a fertilized ovum comes and implants, okay, uterus is ready. But if the fertilized ovum is not there, then we don't want that extra epithelium to be there. So, estrogen and progesterone actually decrease in levels, and there is apoptosis of the epithelial cells. Okay, now let's see what is the pathological significance of apoptosis. See, when inside a cell there is damage of DNA, maybe due to radiation, or there is accumulation of misfolded proteins. Normally, proteins are folded as they are being formed. There is folding of the amino acid chains. But if there is accumulation of misfolded proteins, there is some error, then those cells should undergo apoptosis. Thirdly, if there is viral infections also, this is also a cause that it can cause apoptosis. That we will see how that is happening. So, any damage inside the cell like DNA damage, misfolded proteins, or even endoplasmic reticulum damage, mitochondrial damage, all will induce apoptosis of the cell. So, when this apoptosis starts, there are certain morphological changes which start happening in the cell. So, first of all, see that actually during apoptosis, cell produces proteins which are necessary for self-destruction, and there is no release of toxic substances. Now, this causes some morphological changes like there is condensation of the nucleus, there is a fragmentation of the DNA, so the whole DNA breaks into small fragments, then the cell actually shrinks in size. Okay, what we saw, the cell murder or necrosis, in that the cell swelling occurs. In apoptosis, cell actually shrinks in size. And then the fragments which are forming, so after cell shrinkage, the fragment of the cytoplasm also start forming, and these fragments are known as apoptotic bodies which start getting separated from the main cell and fall off. That is why we said that apoptosis literally meaning is falling off. So, these apoptotic bodies which are released are then rapidly engulfed and digested by the macrophages. They come to the site, they identify them, these apoptotic bodies and phagocytize them. So, here is a diagrammatic representation of the apoptosis. This is showing the normal cell, right, with normal organelles, there is a normal nucleus. But as apoptotic signal comes here, cell shrinkage starts, the chromatin condenses, there is a formation of the DNA fragments, DNA starts breaking. Then you see here that cytoplasmic blebs start forming. So, as the organelles are fragmenting, along with that the cytoplasm starts forming small blebs, and ultimately these blebs separate from the cytoplasm, carrying with them some aspects of the fragmentation of the organelles and the fragments of the nucleus. So, these apoptotic bodies fall off from the cell. After that, these are identified by macrophages which come to the site and phagocytose them. So, there is no inflammation during apoptosis since there are no immune cells at the site of apoptosis, and because of this it is like a silent death, right? Nobody knows in the surroundings that the cell is undergoing cell death, and that is why this apoptosis is not visible in light microscopy. Okay, so how does this apoptosis occur? Well, there are two pathways. There is an intrinsic pathway which is also known as a mitochondrial pathway, and there is an extrinsic pathway also known as death receptor pathway. A part of it is we have already dealt when we were talking about the growth signal absence or the presence of a death signal. We will see in detail in short while. But you see that this aspect that how apoptosis occurs, Sidney Brenner, Robert Horvitz and John Surston have received a Nobel Prize in 2002 in physiology, and their work was on genetic regulation of organ development, that is the embryogenesis, and programmed cell death during that time.